right, so the fourth lecture in this class is focused on the history of the E. coli reconstruction. So to frame it just a little bit, we started out in lecture 1 talking about systems biology in general and the need to integrate data types and the fact that we could do it mathematically if we had proper representation of the chemistry and the genetics. Then in lecture 2, we talked about the concept of reconstruction in a very abstract way. And in lecture 3, we went through the process of reconstruction kind of generically, that it is a workflow, and we talked about some of the steps and some of the issues associated with that. So today, we're going to go through how that workflow was actually implemented to build the E. coli model. And as you will see, this process started before we had the genome, and the first small-scale reconstructions of E. coli came out in the early 90s. So we are now in the middle of the 2010s. So this has been a 25-year process. Okay, so we will go through a few things. First, the history of E. coli as an organism and the role it has in systems biology. As we all know, E. coli has been a model organism for so many things in the life sciences, and systems biology is no exception to that. So the reconstruction of E. coli metabolism has been under development for 2.5 decades, like mentioned, and started before we had the genome sequence, which came out in 1997. And as I mentioned last time, the reconstruction process is iterative, and there have been 4 major iterations of this reconstruction since the sequence came out. And hopefully the 5th one will appear before too long. The current reconstruction that is published is called IJO 1366, and which is now being used more and more. And how that alphanumeric string comes together to name the reconstruction will be explained later in the talk. Most of this lecture, over 80% of the time, will be spent on the history. We'll talk a little bit at the end at where we are in time with it and what may be ahead. And it has now become a platform for systematic discovery of what we don't know. And we'll talk about that, and we'll, of course, mention that more later in the class. One of the things we can do with a model organism now, and a well-curated model, is to map it onto similar genomes, so kind of next of kin. We can map it and actually get really good draft models for related organisms. Then we can start to look at what that organism has over and above your reference, and vice versa. We mentioned this a little bit last time when we talked about the efforts to map Recon-1 onto other mammalian genomes. And that has not been as successful as expected, presumably because the annotation of those other mammalian genomes isn't as good as it should be. And when you start doing that, this issue of what's a strain and what's a species comes into focus, and a few other things. Broadly speaking, we can look at this as a perturbation variable on the model by a small incremental step in the phylogenetic tree. Okay, and the uses of the reconstruction, I mentioned them a little bit last time. I'll say a few more words about that today. So that's the program for this lecture. So here's a quote to Fred Neidhart, who was the editor of those big volumes on E. coli that used to be called the E. coli and Salmonella Bible, where he says that every biologist is really interested in 2 organisms, E. coli and something else. And this is actually very true, because E. coli has become the reference for so many things in biology. So just a few words about E. coli. First of all, it's a gram-negative bacterium that belongs to the Enterobacteria family. And I see that that clip is running there. It shows dividing E. coli expressing GFP in a cell cycle specific manner. That's from Jeff Hasty's lab here at the UCSD. It was discovered over 100 years ago by a pediatrician, isolated from feces, enterobacteria, clearly. And we all have our own E. coli, by the way. If I were to sample your stool and isolate E. coli and sequence it, they will all be slightly different. It's kind of interesting. We are not only different in our own genetic makeup, but also in the makeup of our microbiota. So it's been known for a long time, became a common model, and of course the studies that Jacques Monod did on it and so forth helped to catapult it into this model organism status that it has. It's a very flexible bacterium. It grows on a bunch of different carbon sources, in contrast to, say, some of the environmental isolates we may take out there from the soil, where the microenvironmental niche where growth happens is very narrow. E. coli has a very broad set of catabolic capabilities and can grow with and without oxygen and so forth. When you study organisms, you always have to view them through the eyes of evolution and try to understand their lifestyle to figure out what they're actually doing, what homeostatic states do they strive for, and so forth. So E. coli, commensal E. coli, it lives with mammals mostly, or actually any organism that has a gut, has basically a 4-step life cycle, and each one of them is characterized by a shock. Something big happens. So let's imagine there is E. coli growing on the grass out there or something, and it gets scooped up and eaten by an animal. The first thing that's going to happen to it, it'll experience a heat shock. It'll first go to 37 degrees C or so from ambient temperature. And that's his first clue that something is about to happen. All of a sudden there's a heat shock, and there's a bunch of things in E. coli that sense the ambient temperature. That's then followed by a pH shock when it goes into the stomach. And now E. coli is pretty certain what's about to happen. So the third shock, of course, is to get into the GI tract and ultimately into the colon, where they are going to be in a fully anaerobic environment, very rich nutritional environment, and a lot of competing species, quite different from where they started. Then, of course, there's the cold shock at the end, where it goes back into ambient temperature, plenty of oxygen, and nutrients evaporating away. So E. coli is prepared to go through these 4 shocks. It's programmed to do this. And this kind of physiological assessment through the eyes of evolution to understand the lifestyle of the organism that you might be working with is very important. So there are now thousands of sequence strains of E. coli, and it's quite likely that before too long that essentially every infection diagnosed will have the pathogen sequenced. So this information is becoming very cheap and easy to get. I should say that we here in my laboratory are now getting clinical isolates from the infectious disease ward here, and we can resequence 20 or so strains overnight for maybe $1,200 or something like that, $1,500. So it's cost maybe $50 to $75 to get the specific DNA sequence of a pathogen. And you can map those sequences onto now many, many reference strains. So the E. coli species, there are strains and there are species. There are quite a variety of them. Oops, let me see. There are non-commensal strains that don't grow in association with a mammal or an insect, don't have a host. There are commensal strains that just live in our gut and don't do anything pathogenic. Then there's a series of pathogenic strains that are grow in a variety of niches in the human body. And it was surprising to me to find out when we went to the hospital that E. coli infections is one of the major types of infections that disease units in hospitals in the United States have to deal with. It's Staph aureus, Pseudomonas, and E. coli. It's probably over 90% of the cases they have to deal with. And I believe with E. coli, the preponderance of them are uropathogenic. I think 70% of urinary tract infections diagnosed are E. coli. So E. coli, some strains of it have specialized into a microenvironmental niche growing in the body, but by and large, they are the same strain. I mean, broadly speaking. And here's just a list of strains that's been sequenced, and here's a graph that everybody should see in case you haven't seen it. This is the cost of DNA sequencing per base pair on a logarithmic scale, and that dashed line is Moore's law. Somewhere around 2007, there's this incredible drop in the price of sequencing by logs, within a few years. And this is what has fundamentally changed the life sciences, that we can, for $50, sequence every infectious strain isolated that has infected a human being. Now people talk about sequencing human genomes for, some say less than $1,000, some say $1,500. So it's quite likely that everybody's sequence will become a part of our medical records. But anyway, this is a dramatic change. And being here in San Diego, we all know that Illumina has played a big role in making this happen. Okay, so why model E. coli? I think the reasons are pretty obvious. The bibliome for it is very deep. There's a lot of publications available on E. coli. We mentioned the SKI index last time, this index that has been put out by the European Bioinformatics Institute. E. coli has the highest SKI index, so a lot of literature available for every gene in E. coli, or most genes, I should say. It continues to be a model organism for a variety of things. The collection of omics data types for E. coli is vast. A lot of information coming out, and we'll look at some of the websites that host that here in a moment. It's very easy to grow and manipulate. Everybody in this room with minimal experience can be growing E. coli in the lab within a week. So it's a relatively easy organism to grow with, work with, I should say. And it's very easy to genetically manipulate. It used to be the easiest organism to genetically manipulate, but now, of course, with the development of CRISPR-Cas9, we can manipulate or edit genomes, many more genomes than E. coli's. But I should just note that the knockout system, the easy-to-use knockout system in E. coli was developed in 2000. Prior to that, it was so hard to genetically manipulate any organism, including E. coli. We'll talk a little bit more about that later on. It's model Enterobacteria. There are many human pathogens related to it, closely related to it. We'll talk about that later on. Klebsiella, Salmonella, Yersinia, and others. And then there is this famous quote to Jacques Monod, where it says, anything found to be true of E. coli must be also true of an elephant. And most people just laughed this off at the time, but now we know that the gene content is quite similar between organisms. So detailing what's happening in E. coli is very valuable for a lot of other organisms and some of their functions. Now, the data that has been generated for E. coli and the knowledge has been accumulated, assembled, put on the web. Here is a number of web resources to look at. So if you're dealing with E. coli or enterobacterial species broadly, there's a lot of bibliomic information available to you. So this is, of course, perhaps the main reason why the E. coli model was the one to develop the fastest, could be validated the most extensively, and also put to use the most extensively. Okay, so one of the characteristics of the reconstruction process that I mentioned last time is its iterative nature. You build a model, characterize it, then there's maybe more information available on the organism, so then you expand the scope of the model. And you can cover now more biological functions, so you can study them, compute them, predict them, and so forth. So it's going to be an iterative process, and I just want to walk through the history of the major iterations of the E. coli reconstruction. So first, when I introduce you to the E. coli orphometer, you can build an orphometer for any organism you're trying to reconstruct. It's just a counter that just counts the number of genes that you're representing in your reconstruction. So in 2007, the third iteration of this genome scale model was published, and there were 1,260 genes and their products and their gene products accounted for in the reconstruction. And I'll put that number in context here in a moment. And then a few years later, genes kept being put into a reconstruction, and the orphometer was at 1344 at the beginning of 2011. And we'll see later in the class that this number now is much higher. And it is important relative to the number of functionally annotated genes in E. coli. And I'll talk about that in a moment. Okay, so let's talk about the naming convention. I was lucky enough to meet Rodriguez Bolivar at the trip down to Mexico half a dozen years ago or so. And he cloned a plasmid called PBR322, which used to be a workhorse plasmid in the history of biotechnology. And the name there, this alphanumeric string, has a meaning. So the lowercase p that we start with stands for a plasmid. And BR are his initials. And then there's a number. And I believe this was some version number of the plasmid, number 322. So that's how the plasmid was identified. And this particular plasmid became widely used. So this naming convention became the inspiration for how to name reconstructions. So they often start with an I, meaning in silico. It's not the real thing, it's an in silico representation of a cell. Then the initials, maybe of the person that was primarily responsible for building the reconstruction. If there's more than one, this could be maybe made a little more complicated. So Adam Feist built a reconstruction of E. coli that became widely used, and so his initials are there. And then finally, the count on the orphometer once the reconstruction is published. So this particular one accounted for the function of 1,260 metabolic genes in E. coli. So that's the naming convention. And I think there are maybe 100 or so reconstructions now that out there that have this kind of a name. So, okay, so let's talk now about the different reconstructions and how they were built and what they did, what they could predict, and how they, over time, generated more and more predictions and generated more and more credibility and more acceptance. So, before we had the genome reconstruction, it was possible to reconstruct the basic metabolic pathways in E. coli, fundamentally based on information that was assembled in that E. coli book that I showed you before. And there was a gentleman called Amit Varma that did that first. And then we didn't have a gene count, so that's in quotation marks, but this is the size of the reconstruction and when it was built, so this is in the early 90s. It contained the basic catabolic and anabolic pathways in E. coli. You can compute a number of things with it. You can compute the ATP yield on different substrates, the redox yield on different substrates, depending on what the electron acceptor was available. It allowed us to compute theoretical yields of products in E. coli, like amino acids, and this actually turned out to be quite important. And being able to calculate theoretical yields of potential products like amino acids from reconstructions has been extensively used, because often companies in the industrial biotech space make decisions based on how much yield they can get from the substrate, because that's the primary cost driver in the product in the end. So that proved to be quite interesting. And you could also calculate basic pathway usage for optimal and suboptimal growth of E. coli. We can talk more about that in a moment. But then what could be done here with a relatively simple model that just computed the overall growth and metabolic characteristics, these computations could be compared to actual growth conditions. And this figure here is from a paper published in 93 that was actually quite astonishing. What the figure shows is comparison of computation and experimental data. And the solid lines in here are pure predictions. The model just started to calculate at the beginning once it had been fully parameterized. And we'll get to what those parameters are later in the class. But these are predictions. And you can see growth rate is predicted, acetate secretion and reconsumption is predicted, and a few other things. This is done in a fed batch mode, that is shown to the bottom right, where you inoculate a chamber that is stirred, and you keep dripping substrate in there. So you do provide more substrate over time. And if you look at the graph to the top right, that's the glucose concentration in the media, and you see in the beginning it's going up because there's more glucose coming in than consumed, and somewhere around 5 hours or so it starts going down. If you look at the growth rate curve at 5 hours or so, this is when exponential growth is really taking off, and when the cell density goes up, the glucose concentration in the media goes down and hits zero. So there's so much cell mass in this fat batch device that as soon as you put a drop in of fresh substrate, it's immediately consumed. So not only did this model compute quantitatively the secretion rate of acetate and then the reconsumption, but also predicted accurately which metabolic byproducts were being made. And that's another astonishing prediction. You know, E. coli makes acetate, it pickles itself, as some people say, with time if you leave it alone. Yeast will, on the other hand, make ethanol, and the yeast model predicts ethanol as the best byproduct being produced. And we'll talk later about how these predictions are made, but these are basically predictions made based on econometric arguments on the optimal use of a substrate. So the least useful intermediate is dumped first. So we'll talk about that later. So this was actually astonishing that this could be done. And this experimental study done by a student that had never grown E. coli before, just like maybe people in this room, went in and did these experiments, took about 3 months, and was able to produce these results. You note that the reconsumption of acetate is slightly off because this model doesn't account for the timescales of transcription because some genes have to be upregulated to reconsume the acetate. I will talk about that later. Then there was another iteration of this reconstruction that came out of Berkeley, out of the Kiesling lab, which added more pathways into this model that accounted for, like I said, basic catabolic and anabolic pathways. So it detailed the synthesis of cofactors, looked a little more at the synthesis of the cell wall, and so forth. And this model was used to interpret fluxomic data, labeling data. And the labeling data was consistent with the predicted pathway usage inside the organism. So these are the two reconstructions that were built before the genome came out. So you see this was published in 97, 98. The genome was sequenced and published in 97. And there was quite an interesting history behind the publication of that sequence and why E. coli was beat by Haemophilus influenza. That genome was the first one to be published in 95, as we mentioned before. But anyway, so the E. coli genome sequence for the K12 MG1655 strain was published that year with its annotation. And then Jeremy Edwards, who was a student here in bioengineering, went through the whole annotation of the genome and started picking out all the gene products that had metabolic annotation. So he could take everything out of the reconstruction that was known to be or thought to be associated with metabolism. And that led to the first genome scale reconstruction of E. coli metabolism published in PNAS in 2000. The Haemophilus genome scale model was actually the first one to be published in 99. Okay, so what was this used for? This was actually the first reconstruction that could now predict the consequence of genetic manipulation, because genes were explicitly accounted for in here. And in 2000, there were a few dozen metabolic knockout mutants available for E. coli. And as I show in the next slide, the prediction of lethality of gene deletion, or prediction of gene essentiality under a given condition, was enabled. This model was also used now to design quantitative experiments, including adaptive evolutionary experiments, and predicting their outcome, and did so successfully. And this is something we'll cover later in the class when we talk about adaptive evolution. And these were actually astonishing predictions that could be made this way. Genetic basis of lethality predicted, outcome of adaptive evolution. So it seemed like at the time that genome scale models would become quite useful for a variety of purposes. And work on these genome scale models over the last decade and a half have shown that to be true. Okay. The last bullet point there is maybe worth mentioning too, which was kind of the first assessment of the incompleteness of our knowledge of E. coli metabolism, and this is something we'll call gap filling and talk more about later on. So here is this prediction of gene essentiality that I mentioned. As we'll see later in the class, we talk about objective functions, which are called Z. So an objective function is a mathematical representation of what you think the organism is trying to achieve. And in this case, Z is the growth rate. And this graph shows a scaled prediction of growth rates relative to the wild type. So it's the growth rate of the mutant divided by the growth rate of the wild type. And there are, I think, something like, I forget now, there were a total of, well, it's down in the corner there, there were 79 cases that could be looked at here, and 68 of them were predicted correctly with an 86% hit rate. And you see the first few genes that are, the first few columns in this graph had the names of the lethal genes because there's no growth predicted. Then you had these columns in yellow that predict retardation of growth. And those predictions also proved to be quite accurate. And then there are the blue ones where the gene is completely redundant because essentially the same growth rate can be achieved without that gene. So there are three categories of predictions here. Redundant genes, genes that actually will show a phenotype, and then genes that will show the lethal phenotype. So many such studies have appeared since this paper came out. Then Jenny Reed built a model a few years later. There was a group out of Cold Spring Harbor that published a re-annotation of the E. coli genome around 2001, as I recall, or so, which was an update from the re-annotation published in 1997. So now there was more information about the annotation and the gene portfolio of this strain. So Jenny set now out to begin to put some quality controls into the reconstruction. So this was the first reconstruction that actually had elementally and charge balance reactions. She also expanded the number of transporters in the model. And I mentioned this last time, that the membrane protein, including transporters, are the hardest one to get the function for. And there is a database hosted here at UCSD by Milton Sayer called I think, Transporter DB, that is one of the resources to go to for information about transporters, and she's used that extensively. She also, therefore, was able to look at the catabolic capabilities of the organism much more extensively, because there were transporters now to import stuff into E. coli that weren't there before, the in silico model of E. coli. She also bit the bullet and started looking at the electron transport system. And I mentioned to you last time, this is another process that is very hard to reconstruct. And there was some more information became available on the quinone usage in the electron transport system. And we may talk more about this later, but these are key molecules in carrying the electrons down the chain. And she also built for the first time this explicit relationship between the genes and the protein complexes and the reactions, what we now know as GPRs. So she moved the reconstruction more from a content type emphasis that Jeremy could have to a workflow and quality control type environment. And she could do things like this. You can now predict with full elemental and charge balancing whether growth on a particular substrate in minimal media will lead to acidification of the media or whether it's going to get more basic. And this is a graph that shows such predictions. On the y-axis is just higher and higher growth rate for growth on this is on the substrates indicated. And you see some substrates like glucose that's in gray on this diagram goes off to the right and is predicting a net proton efflux. So the pH of the media should go down. And then there are some other substrates in there like acetate and so forth that go to the left that predict that the pH should be going up. So these are predictions just based on overall elemental and charge balancing. And there have been some papers that have come out subsequent to that show this to be the case. So there's an experiment versus computation. First set of bars there is E. coli on glucose, E. coli on acetate. So acidification above the zero line and getting the media basic below. And then there are some other organisms considered on this graph as well that had been reconstructed and can make such predictions. So here's another type of a prediction you can make. Now here's another one that's extraordinarily impressive. So this is from a paper in 2004. So this is a gene to environment cross. So there's a bunch of genes knocked out and a bunch of environments tested. So as I recall, I think there are something like 120 growth environments on here from amino acid-based, carbon-based, different nitrogen sources and so forth. And then the columns in here are different gene knockouts, all the way from genes involved in amino acid to transporters furthest to the right. So the number of dots on this matrix is, I think, 13,500. I think it's roughly 120 by 110. So this is an astonishing number of phenotypic outcomes being predicted here. And I believe this is actually, and that's maybe why Nature published it, this was the granted scale attempt to computationally predict phenotypic outcomes. And they are growth, no growth. So the growth screens like this are available online now for a number of organisms. So the growth rate data, or the ability to grow or not to grow, was published, and you can compute the outcome of that experiment with Jenny's model, and you can overlay her results onto the experimental results and then color code. And the agreements are in green, and the disagreements are in red. And the yellow dots there indicate whether some regulatory events were included in a model. This was also the first attempt to incorporate the effect of transcription factors that would customize the model to growth under those conditions by not allowing the use of genes that weren't expressed experimentally in the computation. So we'll talk more about that later. But you see that there are many more green and red dots on there. And in fact, I think it's over 90% coverage. And you also see the errors have a particular structure. There are certain columns with a lot of red, certain rows with a lot of red. And it turned out, and Jenny has showed this in a recent paper, that relatively few updates to the model make a lot of these red dots go away. And you can predict now what the cause is for a failure mode. We'll talk about that also in a moment. So this kind of an outcome, this large-scale assessment on the genetic basis for the ability to grow in a given nutritional environment gave a model like this a lot of credibility. It had extensive coverage of the metabolic gene portfolio in E. coli and was able to reproduce that. Okay, then a few years passed and Adam built his model and published in 2007. He scoured the literature for additional genes with metabolic annotation and found 357 that he added in there. He also, for the first time, compartmentalized that model. And we talked about location and its meaning in biology earlier. And one measure of location is cellular compartment. So E. coli has now cytoplasmic, periplasmic, and an extracellular space. And as we'll talk about later in the class, we now explicitly look at the composition of the membrane in terms of its proteome, inner and outer. So the model is getting compartmentalized. In a reconstruction, there are often lumped reactions. So you've probably seen the net reaction for glycolysis, where a glucose molecule plus oxygen becomes 6 CO2s plus water. That is a lumped reaction for all of the glycolytic reactions. And for some processes in E. coli, they have been represented like this, particularly cell wall synthesis. And Adam went through the painstaking process of parsing that out into individual reactions. And this has now become quite important, because now we are starting to look at the ability to build specific models of human pathogens of a particular serotype. So we have to detail the composition of the outer membrane of E. coli and the various strains of E. coli. This was also the reconstruction where thermodynamic information was included extensively. And this was provided by Vasily Hatsimanikaris at the EPFL in Switzerland. We also, at this point, were able to align the contents of this reconstruction completely with EcoPsych, which is one of those resources of E. coli that is extensively used. So this is what he built. He then went through a very detailed assessment of the growth capabilities of this model. And I don't think we need to look at any one of these numbers here, but there are carbon sources there, 325 and so forth. Then there's a column how many he could predict out of that portfolio of substrates relative to what Jenny's model did, just to indicate the expansion in model capabilities. Then there's a column there with the experimental outcomes that you can compare it to. And then what one does in validating models like this, you look at when there's experimental growth, so e.g. computational growth, and when that matches, and when there's experimental no growth and computationally no growth. So these are the predictions that are true. Then there are the errors or the mispredictions. Sometimes you predict that an organism is growing when it's not. So there is some constraining process that prevents growth. It could be transcriptional regulation that doesn't express the genes that the model thinks are available. And then the opposite, which is when you don't predict growth, but the organism grows, which points to some missing metabolic capability in the organism that is not in your model. And that outcome has proven to be a ripe, or I should say a target-rich list for discovering new gene functions, the gap-filling process. And then he looked at the ability to predict the outcome of these knockout screens. And you can see that the diagonal elements here, the accurate predictions are what, 13 plus 78%, so 91%. And the two off-diagonal elements are 3 and 6% failure rates of predictions in these two categories that I mentioned. And this is kind of astonishing that you can actually predict over 90% of these outcomes accurately. And when you fail, you can systematically discover why you failed. So this almost becomes an algorithmic way now just to figure out exactly how an organism works. I kind of draw the similarity often, or the analogy, to studying a language. You know, when you know every other word in a language, you don't understand the speech. But when you understand something like 4 out of 5, you understand what's being said, even if you don't understand 1 out of 5 words. You can kind of gap fill and guess what that is. So that's where these models have come to. I mean, you get a comprehensive understanding, but it's not perfect. So this process continued. Here is Jeff Orth, who published Reconstruction with what ended up being 1,366 genes, not 1,344. He kept accumulating published papers after Adam's reconstruction, and I'll show you a graph of that in a moment. He then systematically calculated using new algorithms called gap fill, gap find, and so forth that have come out of Costas Maranis' laboratory at Penn State. He applied those to figure out comprehensively hypotheses for gap filling, and some of those have been now pursued in the lab since then. And in fact, there was a paper in PNAS a couple of weeks ago based on addressing those gaps. He detailed the biomass requirement a little bit. Remember, we talked about the third stage of the biomass function. We're getting into the prosthetic group requirement and the details. He, for instance, put in the assembly of iron sulfur clusters and how they're needed for certain chemical, some of the reactions. He looked at the growth-associated maintenance and non-growth associated maintenance in more detail. These are strain-specific parameters that need to be measured for your target strain. And later in the class, we'll talk about that fact that we are beginning to be able to reconstruct the processes that are being represented by these phenomenological coefficients. So maybe in time, we'll be able to calculate the energy requirements for these maintenance processes in a cell or in a target organism. Now, his reconstruction also kind of indicated the fact that this was now snapping into a prospective mode. So he started doing prospective experiments, and he put in there a couple of knockout, the screens of a few knockout strains to address some of the questions that came up during the reconstruction process. So now data started coming into the reconstructions being used in a prospective manner. And then his model has been used for gap filling, like I said, and as I'll show you also in a moment, mapped onto other E. coli strains that started, whose sequence started to become available around the year 2010. So here is a snapshot of how the bibliome is growing over time. Of course, that grows much more slowly than high throughput data. So this is the number of references accounted for in his model and Jeff's model compared to the Adam Feist model in 2007. So you see after 2007, there are maybe a dozen or two papers published every year that discover some new metabolic capability in E. coli. And this ends at 2011, and that's actually continued past 2011. There are more and more metabolic processes being discovered. And he really went back through the whole literature, and there is like one paper a year that somehow had been missed in the previous reconstructions, and did a much more detailed bibliomic curation of the reconstruction. And that little pie chart shows the different categories in which these genes fell. Oh yes. Then there is the network map. Okay, there it is. Big image, hard to process, I guess. This is the map. This is the graphical representation of IGO 1366. Pretty complicated. This is where we need a Google map type representation, where you can zoom in and out in different sectors of metabolism. And this is also where we might need automated map layout algorithms to present this map in a manner that interests us. And we may want to show different attributes of this map, depending on what questions we are asking. But right now, maps are available like this. They're just frozen maps, still maps, and you can zoom in and out of them. But this is an area where some work is needed. Okay, so that's the most comprehensive map published to date. And that kind of asks the question, where are we now? After 2011 from 91, 20 years roughly, what did this achieve? And what we have is a genome scale version of this figure I showed earlier, which just shows a reconstruction of a few pathways. This is now available for the entire map I had on the previous slide. So you can imagine that's an incredible amount of knowledge represented by that map, because behind it are the full genome associations, the genome location of these gene products, information about the promoters that are upstream from there, and so forth. And maybe soon we'll have even the protein structures represented in here too, and all the properties that come with that. But that's not the case today. So here is a table that compares Jenny's, Adam's, and Jeff's reconstruction. It's quite detailed, so I don't want to talk much about it, but I'll just point out a few things. One thing that's astonishing with E. coli, that there is experimental coverage of 97% of the open reading frames accounted for in that model. Only 3% of them don't have, meaningful experimental data. So it's a highly curated model. Here's the size of the reactome, which is what they're ultimately reconstructing here. And that count stands now at over 2,200 reactions in E. coli, and those are the 2,200 reactions represented on that map I had there before. And then there's a line in here that shows the non-gene associated reactions. I mentioned these earlier for the lysine pathway. So about 6% of the reactions that are in there are still non-gene associated, and a large fraction of them are transporters. It's often very hard to associate a transporter to the compound that's being transported. And we'll talk a little bit later about, not shown in here, the coverage of structural information we now have for these proteins. This is a visual representation of what is in there in this reconstruction. Not gonna say too much about that here, but the top graph shows the GO categories, all of them, and the number of reactions that is represented in each one of these GO categories. And that slightly shaded tip of each bar are the non-gene associated reactions. So you can see they tend to be concentrated in some categories and not others. This model has been evaluated in detail with this 2x2 test that we talked about before, experimental growth, no growth compared to computational growth, no growth. And these are the outcomes of that shown in that table on top. And the diagonal elements, when you sum them up, are over 90%. Forget now what exactly that is, but 92 or 3%. And the off-diagonal elements being, 6, 7, 8% or so. What is interesting, though, what Jeff did is to show, and this is color-coded too, which GO categories are represented in these boxes. So that green box on top is pretty much the same outcome in all the GO categories. If you look at the off-diagonal elements where the failure modes are, the failure modes they are associated with just a few of the GO categories. So there's, in some of these GO categories, we have less complete information than others. So it helps, focus your search. And the second bar there, P, is the cofactor in the prosthetic group synthesis, which still needs information. He put in the iron clusters, iron sulfur clusters, but there are more processes like this that have to be built. And if you're trying to address them computationally, you may end up with a failure mode. Okay, so this has been a 20-year history or so, and I think I showed this slide before. This is the lecture so far is the material behind this graph. So what it shows is a timeline and the key papers over time. And on the y-axis, we have the serial layering of all the processes that were reconstructed. And if you remember from the second lecture, you can reconstruct a network module by module and then integrate them. And this is what this history represents. And then these are the counters for the metabolites, the genes and protein included in there. And of course, the arrow there that shows the genome era, has enabled all of this, the sequence and better annotation to them. And I think I showed the faces of all these people along the way. Ines to the furthest right reconstructed the protein synthesis network in E. coli that is now in the reconstruction, and we'll talk about that separately in a later lecture. Now, just a couple of comments here at the end. So like I said, there have been close to 100 models now published. And since the E. coli model is so well curated and so well known, the temptation is to start with mapping that onto your organism. And then there will be a lot of work going past that to find the organism-specific capabilities in this non-E. coli organism. That requires a lot of work. So this little assessment that was published about a year ago shows the comparison of the reactome content of these different reconstructions. And this was actually quite telling. So this is basically, you have a presence-absence call for each genome, whether the reaction is there or not. And so you have a binary matrix of zeros and ones, where every column is a new organism. And then you do a decomposition of it, PCA type analysis. And this is the outcome of that. Most of the models are in that little green swath. They're very similar in their reactome content. But there are three notable branches coming out of that core. The orange swath are the photosynthetic reconstructions that have come up. And of course, there's no photosynthesis in E. coli, so this is very different. The blue swath there shows the successive E. coli reconstructions and the enterobacterial reconstructions. So they're becoming more and more specific to enterobacterial content. And that yellow swath shows the yeast reconstruction that has also gone through a series of iterations that we'll talk about later. And that's also separating cell from the pact based on yeast-specific information. I should say that the human reconstruction is not in here. That will be a completely different category. There is a new Pseudomonas putida reconstruction coming out that will generate a new branch in this tree. So this kind of, so I think I should show a blow-up of the green area. It's shown there. So most of the published reconstructions are very similar in the reactome content. And this is just a zoom in on that blue branch that shows the E. coli reconstructions becoming more and more specific. This assessment here leads to a few conclusions that are also stated in this piece there. If we are going to cover the phylogenetic tree with good metabolic reconstructions and get a good snapshot of the metabolic capabilities currently resident on this planet, we have a lot of work ahead of us, but we also know how to do it. And this pentagon, maybe you should look through it to go around counterclockwise around it, like that blue arrow shows. The first thing is you have to go through the hard work of accumulating the organism-specific information and put it into the E. coli-based initial reconstruction, which also may lead you to throw out some E. coli-based content. You just have to do this. This is possible for Pseudomonas, Klebsiellas, Streptomyces species, and there are a few other species where there's a lot of information. You need to do this. And probably don't take the time to walk around this diagram. You can just take a look at it. But we now have kind of a definition of what the global gaps are to get this going. And like I mentioned earlier, I think that there's a large-scale project ahead of us here to try to elucidate all of this. This is one of the grand challenges in biology, is to map out all metabolism that is currently carried out on the planet. Probably a 10-year project. Okay, so let's finish up here by talking a little bit what's ahead for the E. coli reconstruction. And again, BENG stands for biochemically, genetically, and genomically structured knowledge base of E. coli. So we now need to go through what's missing in metabolism and the missing gene functions associated with that. That is now a systematic process. And this is focusing not so much anymore on big pathways that are missing, but enzyme promiscuity, this notion of background metabolism, and a few other issues like that, kind of a cleanup operation. There is the reconstruction now of other cellular processes that couple with metabolism. I will talk about those in the class. There is the transcription translation, the whole protein synthesis machinery that now has been reconstructed. This is what Ines Thiele did, as I mentioned earlier. We now have enough data to build the transcriptional regulatory network in detail. There is maybe some conceptual development needed there on how to represent those events. I personally think that they can all be represented by bona fide chemical reactions, but there will be many of them, and we can do that now with all the experimental methods we have available. The two-component signaling systems in E. coli, so the basic sensing mechanisms and how it reacts, can also be reconstructed. There are 20, some of those, 22 or 4, I forget now what the number is. The function of the small RNAs can be reconstructed, but we probably don't have a lot of information on those available yet. There is more of an experimental discovery process needed there before that can be reconstructed. I mentioned that this is ripe now for the inclusion of protein structures, and that opens up a lot of possibilities we'll talk about later. And now the expansion of scope is not so much, this strain of E. coli, but looking at other related strains and looking at what is called the paleo, is trying to figure out what the core processes are that take place in every bacteria. And that's called the paleo, and will be introduced later to what's called the mini-me. So now it's no longer a strain, but we start to look at maybe a species or families of species and so forth. And there are a couple of things here at the end. As you know, there were many spectacular intellects associated in the establishment of molecular biology early on, people with a lot of vision and perspective. And this piece here was written by Francis Crick in the early 70s, 1973, where he realized, and Sidney Brenner is cited as the inspiration for this piece, and I've actually talked to Sidney Brenner about this piece. So Francis Crick wrote this piece called Project K, but now I know from talking to Sidney Brenner, there was also Project L, Project M, and N, the nematode, the mouse, and I forget now what L was, some plant species. But they knew at the time that when they had figured out the basic mechanisms of molecular biology, that this had to be done on scale. But they just didn't quite realize at the time how large that scale was. And it wasn't until the mid-90s when we could start to operate on a genome scale. And the first genome scale model by Jeremy is kind of the first embodiment or the realization of this vision, or this articulated in this paper. You should try to look it up and read it. It's only 3 pages, but it's just remarkable what the leaders of the field of molecular biology operated at that time and how broad their thinking was. Okay, I wanted to end up here with talking a little bit about this opportunity to do the next of kin. And this will take maybe 5 to 10 minutes, because this is an outcome of a term project that was done in this class in 2011 by John Monk. He was sitting in your seat 4 years ago, and at that time there was this flow beginning to take place of fully sequenced E. coli strains. So they were becoming available. And at the time there were 55 of them fully sequenced. Now there are thousands. And the question came up, can the E. coli model that's been built through this 25-year history of a lot of work be useful for building models for strains that are very similar? So this is like a perturbation in genome space to the kind of next of kin in the phylogenetic tree. Is it possible to map these models in a productive way? And so we now have not only sequences for the these E. coli strains that I'll talk about in a moment, but there are also now reconstructions of closely related species, Salmonella, like I mentioned, Klebsiella, Yersinia, which are also enterobacteria and have a large overlap in genomic content with E. coli. So these were also reference models that could be used in this mapping process. And this diagram here shows the, what should I say, the results of the mapping. So you map the genome, you map this reconstruction onto another genome. So you find some fraction of those genes on that genome. Not all of them, but the vast majority of them. But then you find some more metabolic genes on those target genomes. And so you have what's called a core genome, what is common to all the strains of that species. Then you have a pan genome, which is the pieces of the genomes that are customized for each strain. And that's where the pathogenicity islands are, for instance, and some strain-specific properties. And this, the bar chart there is also a categorization of the content, and these are all different metabolic processes represented by this different bar. And the bar on top, the two bars on top, where you see the biggest yellow swath, that's the pan genome, show you which metabolic processes are the most diverse amongst the E. coli species. And these are the catabolic properties and the transporters. So there's quite a bit of diversity amongst the E. coli species in terms of what they can live on. But once they have that stuff imported, the rest of what they do tends to be very similar. So that seems to be the basic discriminating factor in terms of metabolism. And what is remarkable with that is that you can use this to do a machine learning classification, and you can find the discriminators or the classifiers that classify the different species. And that's shown in this diagram over here. And there are a few substrates in there, like taurine and some odd substrates that actually classify these models for you. And then if you look at these substrates that are particular to a category of species, they correlate with the microenvironmental niche where they grow. Not a surprising outcome in the end, but it's surprising you can do this. So like the uropathogenic strain can utilize citrate and some other things, urea, that the other E. coli strains cannot. So you could actually predict here physiological properties and properties basically of the microenvironmental niche in terms of the nutritional environment that these strains are likely to colonize. The other prediction that came out of this, which is impressive, is the prediction of oxotrophies. So all these strains have missing content, the missing metabolic content, and that led to the prediction of oxotrophies, that there were some compounds they needed in the media, and 12 of these strains were available and were grown in different media, and those predictions turned out to be 80% accurate, which is astonishing. All you need is the genome sequence of another strain, and you can predict their properties, preferred growth environment, and oxotrophies. And this came out of a term paper here. He had the basic results at the end of the quarter. Not experimental, but the computational results. Okay, so this diagram is actually a repeat of last time, so I'm not going to talk about it, but the E. coli reconstruction has been used for a number of things. What I wanted to really show here is this diagram, the lower row. So this is the cumulative number of scientific studies that have been enabled by reconstruction. It's widely disseminated. People can download it. People can download the Cobra Toolbox and now Cobra Pi, and can be computing with these reconstructions very quickly. And you see literally hundreds of papers are being generated by these reconstructions from around the world. And the second diagram there shows the growing number of papers that use this reconstruction to drive experiments, not just computational assessment and studies, but actually experiments. So it's becoming, a reconstruction is becoming a tool that is a part of an experimental inquiry. And this is, of course, ultimately where the biggest impact is going to be felt. They can be used, of course, to generate understanding of how an organism works, but one of the practical uses of them is to drive experimentation prospectively. Okay, I am a couple of minutes over, but let me just run through these bullet points here quickly. So the E. coli reconstruction is now highly curated. It's based on multiple revisions of the annotation of the E. coli genome. When I say E. coli, I'm talking about K12 MG1655. Massive number of data sets, very deep coverage of the bibliome. And you saw what Jeff Orth did in kind of trying to get the remaining missing pieces of the bibliome, take it into account, at least up till 2011. If you look at the coverage, these metabolic genes are about 30% of the 400 open reading frames annotated on the E. coli genome. About 700 of the open reading frames have no annotation whatsoever, called Y genes. And there's another 700 Y genes where there is maybe some annotation, but it's not really functional annotation. So the number of functionally annotated genes in E. coli are probably 3,200 or so. And this is about 1,400 genes that are now in the metabolic reconstruction. So almost half of the genes in E. coli are accounted for there. And the vast majority of them have experimentally validated functions. So we are approaching exhaustion of the bibliome, and with a model like this, there is a number of applications we can pursue, and we mentioned them last time, and we'll do a separate lecture on each one of them later in the quarter. But maybe in the grand scheme of things, this reconstruction is the fulfillment of the vision that was laid out in Project K some 40 years ago. Which is a kind of an interesting historical context into which we can place that reconstruction. So that is the end of the history of the E. coli model. Thank you for your attention.